citric acid cycle. Now, when we look at metabolism, traditionally, in almost every textbook from things that you look in high school all the way through college, the biochemistry text, normally the cycles are this. Glycolysis, I start with an energy molecule, sugar. Glycolysis feeds into the citric acid cycle, which will convert those carbons into energy pools, different molecules that have energy involved in them. And then we will go to oxidative phosphorylation, or the electron train, and we will convert that to ATP. But what is really going on inside the body is we have three sources of energy that are coming in: sugars, fats, and proteins. And so what we do is we almost allow us to think that the only energy source is sugar, because we put these three together. And this is how this book puts it together. Then afterwards we're going to look at proteins and fats, how they enter in. But really we're degrading sugars, we're degrading fats, and we're degrading proteins all at the same time, feeding in really to this hub, the citric acid cycle. And the citric acid cycle is the workhorse in turning almost all of the molecules that we eat as fuel sources into usable forms of energy. So this particular chapter becomes even more important because from every pathway that we look on from this point on, whether it is synthetic or degradative, will interface this citric acid cycle almost exclusively. A few will go into the glycolysis, a few will go into some other pathways that we'll look at, but almost everything comes out of here. And so these structures and this cycle is very important. The citric acid cycle is an oxidative degradation process found in eukaryotes and prokaryotes. And so as you can see, we have carbohydrates, fats, and amino acids are coming into this. The carbohydrates have to go through the pathway of glycolysis. But look at those arrows. They are double-headed, and so they come in and out of the citric acid cycle. Also, the citric acid cycle has an AKA, also known as the tricarboxylic acid cycle, or the Krebs cycle. Krebs, from Dr. Krebs, who reviewed this and actually published the cycle first. In terms of the cycle, just like I said, it's both catabolic or degradative and anabolic or synthetic. And so a lot of these different intermediates will be interfaces. When I teach a pathway or I teach how things enter in and out of metabolism, I will teach what's up to new until I get to a structure that is in one of the pathways that you know. And so I'm going to show you a little bit of the road until you get to the highway, because I've already shown you the highway. And so this one is something that you're going to have to remember, because we're going to enter in this particular cycle and out of this particular cycle. One key about glycolysis and citric acid cycle in terms of their relationship: glycolysis occurs in the cytosol of the cells. The citric acid cycle occurs inside the mitochondria. So they are separate and distinct by a membrane. And remember, a membrane's a barrier. So we're going to have a controlled mechanism to get the substrates into or from one compartment to the next. That will include every single most of the energy molecules. At the end of glycolysis, what were the three energy molecules that we had? Pyruvate was one, NADH, and then ATP. The only thing that doesn't have to get into mitochondria is ATP, because ATP is used in the cytosol. Okay? But the NADH and the pyruvate have to get inside. We're going to find there's a problem with the NADH that's going to frustrate our calculation of how many ATPs can be made from one single glucose. And we'll get to that in the next chapter. But the pyruvate definitely has to get in. Now, it is an oxidative degradation, and so we're going to do a lot of redox reactions. And within biological systems, there's a lot of different ways in which we can have redox going on. We can have metal centers move electrons, and usually these are single movements. We can see hydrogens move around that can accept electrons. We can actually look at a movement of a hydride, which is probably what we're going to see most often in our metabolism, and we can react it directly with oxygen. Reacting it directly with oxygen really requires an extraordinary reaction. It involves metal centers. It talks about coordination chemistry. If you took G-Chem with Dr. Krebs, he loves a coordination chemistry with metal centers because he's an inorganic chemist. And most of the other teachers don't get to that section, but he really wants to get to that section because that's his baby. So you may have actually seen some coordination chemistry. I don't normally go into that because it is quite complex, and if you are going to study that in grad school, you'll learn it yourself. But we will see reactions directly with oxygen. And you can take a R group, a methyl group, and add a hydroxide in a very complex reaction. When we take a look at moving electrons, it goes way back to the Nernst equation, so we're way back in GChem 2 again. And so you can calculate changes in energy by looking at standard reduction potentials, the electron acceptor, the electron donator. And so we can do these calculations, and you've done this in GChem 2 before. One thing that we really need to look at in metabolism and understand is once we have the change in energy, we can estimate delta G by using the Nernst equation, the calculation below that has negative n, the number of electrons, Faraday's constant, times that change in electron potential. And in that, most often on a standardized test, this is where you mess up. A hydride has 2 electrons, and people forget to put 2 for the n in this type of calculation. Very simply, instead of an R there with a circle, it should be an arrow for a equation. Pyruvate, if we took pyruvate and we reacted with NADH and we allow the hydride to leave NADH and attack the pyruvate, we create lactic acid, okay, and the NAD+. The 2 half reactions are listed below, and if we take the acceptor minus the donor, we get a numerical value. Placing it into the equation, we come up with, hey, that's very energetically favorable. Now, on this particular calculation, it assumes 1 molar concentrations, and so it's an estimate of the standard. But this reaction would be quite, it would be spontaneous because it gives off so much energy. A calculation as simple as this could be asked on the exam. And so we're looking at, as long as I give you the half reactions or I give you the delta E, you should be able to calculate the amount of energy. What I really want you to understand is how NAD plus and NADH are related. Okay? Now, this is interesting. If you take a look at the structure of NAD plus, okay, it's an electron acceptor. We've already seen it in glycolysis. We're going to see a lot of it in the citric acid cycle. Look at its structure. Adenine, ribose, phosphate, phosphate. If I took this part of the molecule right here, what is that? What does that represent? ADP. Okay. Now, that's kind of, okay, so ADP is part of ATP, and that's energy. And this ADP is part of an electron carrier. I'll show you something later in a moment. Now, that is just kind of carrying things around, all right? There's another ribose, and then this right here. That's a vitamin B, and this is where the hydride is accepted. And the nitrogen has a positive charge within this ring. That is why it's NAD+, that positive. Okay. A hydride will come in, as a leaving group, and it will be accepted at the top of the ring. Now, problem is carbon has 4 bonds, and so when I attack that ring that is aromatic, this pair of electrons has to move, and then this pair of electrons has to move, and the electron pair ends up on the nitrogen, and we now have 2 hydrogens at the top and a lone pair on a nitrogen. I changed the nomenclature to NADH. It has an H, and there's no positive. That's the reduced form. Now, to let go, we have a reverse where the electron pair kicks down, this electron pair goes up. I recreate aromaticity, which is very stable, and a hydride falls off. And so I can put on a hydride and I can take off a hydride. What tends to happen is that the hydride that is accepted by the NAD plus is the one that leaves. The hydrogen on that particular carbon always stays there. Now, in a lot of reactions, we use NADH or we use NAD plus. And so when we want to monitor a reaction over time, we can simply use the UV range of an instrument to look at how much is made or how much is used by simply making a measurement non-destructively. If you were going to track either the starting with none and making more and more during the reaction course, or starting with a lot and it gets used over the course, what wavelength would you determine would be the best wavelength to track or monitor the creation of NADH or the use of NADH? If you had to set up an experiment, where would you measure? 260. Okay, 260 could be an option. Now, 260 has the advantage that you're going to get the best absorbance, the greatest absorbance, okay, of both of the amounts, okay? Now, if you get an absorbance at 260, okay, then you're going to get somewhere, a sample, you're going to get an absorbance that combines both NAD plus and NADH at the same time. Now, my graph here shows 2 separate experiments, one that has NAD plus and then an experiment that has NADH, and then I put them on the graph. If you mix them up, you will get one line, okay, and the line will, at that 260, will be between, somewhere between the bottom and the top line. But the amount of absorbance, you don't know exactly how much comes from NAD plus and how much comes from NADH. You're just going to get a line somewhere between those 2 values. And so, yes, it's the most sensitive place on the graph because it gives the greatest absorbance, but it's hard to detect between the 2 versions. So, what would you choose? Correct. This one, if you have absorbance at all, then you know you're only getting it contributed from the NADH. If there's no absorbance at all, you must have NAD plus, or your lab partner didn't put something in the tube, and you just got water. So, the best differential that you have is at 340, and one of them absorbs absolutely nothing at that range. So that would be the most sensitive place to distinguish the 2. Now, what's good about this? It's a wonderful question on the slide, but if you're going to monitor the reaction, you're going to look up someone else's protocol, and it would have told you to measure at 340. So you don't have to make a guess at these most of the time when you're doing research. I just wanted to show you why we would have selected that if you didn't know. FAD going to FADH2 also accepts a hydride. But this is weird. A lot of people ask me, well, NADA plus only picked up one proton. Why did FAD go from FAD to FADH2 if I accepted only one hydride? Well, it's because of the ring that accepts the hydride itself. Now, check this out. What is that right there? ADP. Okay. That's interesting. And then the ring structure above, which is also another vitamin, is where we can have the acceptance. Now, this particular molecule can accept a single electron at a time, and it also can accept a hydride. We are going to see it most often with a hydride. So if I have a hydride come in and attack, it's probably going to attack there. If that occurs, this happens, and then this can pick up a proton. Okay? That seems to be a possibility. There's also another possibility. A hydride comes in and attacks there. The electrons could go there, and then these pair of electrons could pick up a proton. So you got blue and you have green. And they end up being the same final product. Tell me or give me a rationale. What do you think mostly happens? Is it green or is it blue? Okay, you got a 50-50 chance on that question. Green or blue? Okay, you choose blue. What do you think? Green? Blue? Green? Blue? We're like half and half. Blue? Green? Blue? 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 That was a very weak blue. What? Blue. All right. A hydride is coming in, right? It's attacking. The concept that you would try to think about in this is, all right, you got to align this thing inside the enzyme, right? Which one is more accessible sterically? Okay? If you think about green, here's the problem with green. You got all this stuff right here. And so this is sterically hindered. There's stuff around it. Whereas on the blue, that's totally open from either side. And most of the time, the hydride coming in is accepted at the blue, or represented by the blue arrows, because it is much less strained in terms of sterics. Okay? And what happens is that extra proton that is actually accepted, the FAD takes one hydride and one proton. And so there's only one pair of electrons that is accepted. NAD plus and FAD are normally used in different ways in terms of enzymes. Remember, the FAD usually oxidizes a hydroxide to a ketone. The FAD is normally used, but not exclusively, but normally, to take a straight chain alkyl and put a double bond in. So an elimination. Both of them have the exact same pair of electrons, but the way that the body processes the potential energy that is there, FAD will produce much less ATP than NADH. We'll look at this again later in another chapter. For every NADH, you have the equivalent of 2.5 ATPs worth of energy. And for every FAD that is produced, you have 1.5 ATP equivalent energy. And so they're used differently, and I can extract energy and convert it into ATP differently. All right. Pyruvate is what we ended up in this glycolysis. And the citric acid cycle is the next cycle. But what happens is we've got one step or one enzyme intermediate between the two pathways. And for items to enter into the citric acid cycle, I have to change pyruvate into acetyl-CoA. What this process will do will be an oxidative decarboxylation. Pyruvate, when we look at the structure, has 3 carbons, and acetyl-CoA has 2. Where does the extra carbon go? The name of oxidative decarboxylation, our carboxy group is lost, and that will go off as a CO2. Okay? And so we'll lose an atom of carbon. Once you have reached carbon that is attached to 2 oxygens and double bonds, that's as oxidized as you can go. I cannot extract any more energy from it. And this is the link between the 2 cycles. It occurs in the mitochondrial matrix. So even that steps inside the mitochondria. So we've got to get the pyruvate in. We will do that through a pyruvate proton symport. Oh my goodness. I'm using words that I think you know. Okay, what in the world is us? If I say symport, what do you think about? It's transferring in. It is a protein, okay, embedded in the membrane. And the pyruvate and a proton both bind in and are transported into the cell together, symport together. Okay? Now, what happens in this, is this passive or is this active transport? Could you tell? Do you know? Is it passive? Okay. Normally, passive transport is just the molecule itself. This is actually active, and it is what's called a secondary active transport. Usually, inside the mitochondria, the concentration of pyruvate is high, this is low, and usually the concentration of protons outside is high, and inside is low. And so what I do is I couple the proton gradient to help power pyruvate coming into the cell. All right. Once pyruvate's on the inside of the cell, we can convert it into acetyl-CoA. This is done by an enzyme complex called the pyruvate dehydrogenase complex. And it's a wonderful name because it's straight up. It is a complex reaction set. Now the complex, what they're really talking about is it is 3 enzymes that are placed together. They actually bond or actually interact together and work as a team. All right. Pyruvate dehydrogenase. Dehydrogenase is an oxidative. So that's going to move electrons. Usually the dehydrogenases have an electron carrier associated with them. We have dihydrolipolyl transacetylase, we'll see what that happens, and dihydrolipolyl dehydrogenase, another oxidative process. The net reaction looks very, very simple. Pyruvate plus CoA plus NAD plus yields acetyl-CoA plus CO2 plus NADH. It's not that simple, but it sure does look it. All right. Not only do we have 3 separate proteins, we also have cofactors for many of these. Okay? NAD plus is a cofactor, so it is a small organic molecule that has to be bound to inside the enzyme for the enzyme to function. So there's some other cofactors that are important to play. This enzyme complex has 60 different polypeptides, 20 of each of those versions that are bound together. The size is 5 million Daltons. What does that mean, 5 million Daltons? Again, I'm using something I assume you know, but I don't know if you know. Biochemistry uses a term called Daltons. It's not something we learn in general chemistry, but it is related to something that we learn in general chemistry. Daltons are referring to mass. And in G-Chem, the molecular masses are in what units? What is it? Something with moles. What else? Grams. Man, I am really going all the way back to G-Chem 1. Whew. You know, this integrates. Okay, so a Dalton is really a fancy word to say grams per mole. So a mole of this complex requires 5 million grams. Relatively, this is huge, okay? Very large. The transacetylases form the core, and the pyruvate, the 2 dehydrogenases are bound outside. What's quite interesting about it, when we take pictures with electron microscopes, you can actually see a crystalline structure, a very regular repeating structure in that. That is quite interesting. Now, why in the world do we have a complex like this? Well, it integrates the pathway. So it really is a little tiny pathway itself, but it all happens inside this core. And so we have several reactions that occur. It increases the rate of reaction and the specificity, so we don't get side reactions. The other thing about the pyruvate dehydrogenase complex is the fact that this is so fundamental to the production of energy that we want them to run as fast as they can, and having this large complex can move a lot of pyruvates into acetyl-CoA very quickly. The other cofactors that are important, TPP, thiamine pyrophosphate. Thiamine is a vitamin B, and I show you the structure. The pyrophosphate is what the body converts the thiamine that you take in as the vitamin B into the active species. TPP will be used in a lot of enzymes. This is the first time you see it, but this is not going to be the last time you see it. Lipamide is another cofactor. I'll show you in just in the next slide. We will use FAD, although you don't see it in the reaction because it is used temporarily and then regenerated. You'll convert it into FADH2 and then regenerated. NAD+, you have seen and know about that is in the reaction. And then the CoA is something you may have seen. I'm going to show you what that looks like. The lipamide is a long cofactor that has a very long alkyl chain right here. And lipamide is always, it actually starts off as an acid, but it reacts with a lysine that also has a long chain. And so what you get is this very long tether bound to the enzyme, this is E, and this over here, the lipamide, is the cofactor. The reactive species of the cofactor is right here. So that facilitates the reaction. But the chain is important in transport. So what's going to happen is I'm going to load the reactant on that ring. I'm going to react it with those sulfurs. But that long tail in the middle means that it will react in one active site of the complex, put the molecule on, and then that tether can move and it flops over to the other active site for the next. There's really there's not even an actual diffusion. It actually gets moved, that reactant gets moved from one active site to the next, and that chain can go back and forth. This increases the rate of the reaction and controls. And so you can see after the reaction, we're going to have the carbons, the two carbons attached to the ring. I'll show you what happens in a moment. CoA, you're going to see a lot all over the place. A lot of times we will write it CO with a capital A, and then a lot of times people will articulate dash S dash H. Really, it is, we articulate that, and we say CoA is the rest of it. What did I just circle? ADP. There is one theory that the original biologically active molecule first developed was RNA. We know RNA can hold genetic information. We also know that RNA can be catalytic and an enzyme. It's not as efficient as the enzymes that proteins make, but it can catalyze a reaction. Also, all of the important carrier molecules, it seems, has a piece of RNA in it, including energy, ATP, the electron carriers, the CoA. And so the theory goes that the first biologically active molecules were RNA. It could do everything: genetic, informational, catalytic, and carrier, and power. And so it's an interesting idea, but it looks like the RNA structure is a very fundamental structure throughout all of biology. And so just about everything has it. Now, this CoA, it is like an address and a stamp. When things get attached to that sulfur on the end, CoA usually brings whatever is attached into the degradative process. And so when we are carrying things, part of the active sites that take things in for degradation have a pocket for the CoA. And that's going to be a common thing. Now, we write CoA, and it's really tiny because that's efficient when we put together text. But that CoA is really a very large molecule inside the body. And so it's almost kind of a misnomer, the way that we write it really, really tiny, it representing a very, very large cofactor. All right, so the actual complex is a series of reactions. And so the thing that you should recognize is this right here. That's pyruvate. It was brought in across the membrane, and we're now in the mitochondria. Now I'm going to show you a much, much more detailed process in just a minute. The TPP cofactor is going to be reactive, and we're going to react the TPP. It's going to lose the CO2 here, and it's going to transfer 2 carbons here onto the TPP. That will eventually become an acetyl group. The second reaction is the lipamide that has the sulfur ring, and the sulfur ring is going to be reacted with this carbon to create the acetyl group attached to the sulfur. It will move to another active site that will allow this acetyl group that's attached to a sulfur to react with CoA, which is a sulfur. Now, from the perspective of the acetyl group, it's attached to a sulfur here, and after the reaction, it's attached to a sulfur here that really has no difference in terms of energetics. It's an equilibrium reaction. But now, this acetyl CoA is mobile and can float inside the mitochondria, and now I have a lipamide. But this lipamide is reduced. Okay? This reduction here prevents it from being an active reactant in the next round. So what we have to do is go back and reduce this to close the ring. And so we will take a proton, a hydride will fall off. It's accepted in an FAD first, but that is bound inside the enzyme, never leaves. What is mobile is an NAD+. We'll take that hydride from the FAD, and this will be the mobile. So this is a product, this is a product, and this is a product. Everything else in the formation are regenerated, created and regenerated. And so If we go back to Hess's law, if they're on both sides of the reaction, we cancel them out. That's why the reaction looks so simple at the beginning. But there are a lot of intermediates. Here's another look at that, what enzymes do what reactions. And so the first enzyme is the decarboxylation. The second enzyme is the transfer of the carbons to the acetyl-CoA. The third enzyme facilitates the formation of the NADH. All right. This, no energy, okay? This right here represents energy source that I still can extract energy from. And here I've created another pool of energy. So at this moment in time, we've made a little bit of ATP, but what we're starting to do is make lots and lots of NADHs. All right, and we still have some carbons that still can be oxidized. This enzyme can be controlled through a phosphorylation event. If it is phosphorylated, it is inactive, meaning that I have a change in the structure and I can't fit into the active site. Yes?
So you get 2 NAD.
All right, but if you think about stoichiometry, if we go all the way back to glucose, we'll have 2 pyruvates to create 2 acetyl-CoAs to create 2 NADHs at this step. Okay. In terms of regulation, what things make this go, what things make this stop? All right. If by chance we have a lot of NADH, a lot of acetyl-CoA, a lot of ATP, we stop this enzyme. Okay? Because that's the product of the process that we're going to step into. And so those are all feedback inhibitors. And they regulate only the enzyme 1. If I have a lot of NAD+, that means I can accept a lot. If I have a lot of CoA floating around that has nothing on it, that feed forwards. If I have a lot of ADP, meaning I have a little bit of energy, I tend to make this go. Okay? And if I have calcium, I make this go. Now, why in the world would calcium be a regulator? I can understand intermediates that are part of the process. If it's low or high, that would make it go. I can understand ATP, to make it go. But why in the world would calcium? What do you think about calcium? What is calcium release an indicator of in biological systems? It does let glucose in the cells, yes. It really is in, let me give it a little more, in muscle cell. What does calcium do to muscle cells? It does, but what else? Contraction. And what does contraction need? Energy. And so if the cell sees calcium, well, remember I tell you that most metabolism is taught from the perspective of muscle cells. That's indicating I'm about ready to use a lot of ATP. That means the processes that make ATP, it's going to upregulate. So you'll see calcium quite often as a regulatory molecule in metabolism. All right, here's the mechanism of the pyruvate dehydrogenase complex. The first reaction is the enzyme will react with TPP itself. And that enzyme will deprotonate the proton on the ring structure. And so I'll have a negative carbanion right next to that positive nitrogen. Now, that carbanion will attack the pyruvate. And so I attack at the carbonyl. When I attack at the carbonyl, the electrons kick up. Now, in general, you are seeing one of 2 things. We've had a nucleophile attack a carbonyl. Okay? One option is to kick up, and we've seen that a lot, and then to kick down, and then we have some sort of leaving group. That's one way that nucleophiles attack a carbonyl. We're now seeing a second option. A nucleophile will attack. The electrons can kick up onto the oxygen, but then attack a proton. It becomes a hydroxide. So we can do one of two things at this moment in time. We're actually going to find at least a third one later. Every time that a nucleophile attacks a carbonyl, it could kick up and kick down and have a leaving group fall off, or it can kick up onto the oxygen temporarily and pick up a proton to become a hydroxide. And that's a fairly stable intermediate. Okay. And so we're going to have an intermediate. Now, that CO2 hasn't fallen off yet. It actually falls off in the next step. What happens for it to fall off is that in this form right here, this acid can convert into a CO2. Now, I'm going to articulate something real quick. The lone pairs. And with a negative charge, that means I have 3 lone pairs. Okay? To remember the Lewis structure of CO2, which I know everyone can draw, okay, it represents this. So if I take, that doesn't look like a pair. If I take one pair of those electrons and I move them into a double bond, then I'll have at the top 2 lone pairs, 2 lone pairs. There's where CO2 comes in. But this pair of electrons has to leave. Now, every acid, organic acid, has the ability to be decarboxylated. But it doesn't happen too often because that loss of that CO2 usually creates an intermediate that is very high in energy. But once I put this cofactor on, it allows us to create an intermediate that is lower in energy. So this is why the CO2 falls off. The electron pair forms a double bond, and this electron pair then can sit up onto the nitrogen, going from a positive to a neutral. That's low in energy. And so I create an intermediate where CO2 is lost, and I have a resonance structure. Now, what I'm showing here, I should please draw this arrow. This is a formal resonance structure between the two. You have the one where the lone pair is on the nitrogen, and then you have a lone pair that sits on that carbon to create a carbanion. All right, just for grins and giggles, you have A and B. Where would the equilibrium lie? Which one's a whole lot more stable? A or B? A lot. Like 99% of the time, it's going to sit over in A. Now, it's happy in A. It's not a high energy state, but it's not productive to us. Now, 1% of the time, it will be in the carbanion state, and if it's there, it actually will then react with the lipomid. And so I'll have 0 concentration, the equilibrium will move, and I can drive the reaction all the way through. And when that lone pair attacks one of the sulfurs, the bond between the 2 sulfurs will then pick up a proton. Okay? Here's the problem. I got a bunch of stuff. I got 2 carbons that I'm trying to transfer over. It's attached to the lipomid, and it's attached to the TPP. The enzyme will deprotonate the hydroxide, creating a ketone. That's great. We want that. That will allow the TPP to fall off. That regenerates the catalytic cofactor. That creates acetyl-CoA attached to the lipomid. Now, that lipomid has the CO2, or it has the acetyl-CoA, and then flies over into another active site to allow the acetyl-CoA to react. A base will deprotonate the acetyl-CoA. A sulfur will attack at the carbonyl, up, down. The leaving group in this case will be the lipomid. I have gotten what I wanted. And then I have what I don't want. I have a reduced lipamide. The enzyme will move to the third enzyme. It will deprotonate one of the sulfurs. The electron pair will form the reduced ring, and a hydride is a leaving group accepted by the FAD. Then the FAD will transfer its electron pair to the NAD+, and the NAD+ is mobile. The FAD is always bound inside the enzyme number 3. It never diffuses. And I've created my 2 molecules. All right, all that work because I'm trying to get to the citric acid cycle.
You're saying something's mobile like that. What do you
Yeah, when I say it's mobile, so inside the enzyme, the FAD is sitting inside the active site and never really comes out. So when it gets converted to FADH2, it's still sitting there, and then it'll give the electron, but it never leaves. The NADH comes to the enzyme, docks into the active site, the NAD+, and then when it's NADH, it can float back into the matrix, and it's mobile, moving around. And so that's the difference. Yes?
Where did the other hydroxide or hydrogen come from on the FAD?
On the FAD itself, where did it come from? If we go back, a lot. Okay, next time, stop, there. When the hydride comes in and attacks the blue, the proton comes from inside the enzyme, one amino acid that becomes an acid and can donate it. And that's most of the time what happens. Or if it gets out into the cytosol just from the buffering system, it can pick up the proton. And so, but inside an enzyme, there will be an amino acid right at that position to donate that proton for it to occur. Now, when the things move out, when all the reactants move out, the cytosol comes in, and that amino acid will reprotonate from the cytosol eventually. So eventually, the protons all come from the buffering system within the cytosol. So now we have the citric acid cycle. Stop. It's not as many reactions as we have for glycolysis, but depending upon what you look at and which textbook you look at, do you see there's a reaction 2A and 2B in this cycle? Sometimes they don't show that intermediate. I do like to articulate it because it's important when you're going to do your quiz tonight. Because 2A is a dehydration and 2B is a hydration. So I take off water, I give water, so it kind of equals in terms of the reactants out, but we have a change in the structure itself. Up at the top is the acetyl-CoA, what we just produced. And again, remember from the stoichiometry of 1 glucose, we have 2 of those. And as I go through the cycle, I will produce a CO2 and I will produce a CO2. The 2 carbons that come in from the cycle, okay, we net lose 2 CO2s. And so what we have is a 4-carboned structure. After it condenses with acetyl-CoA, I have a 6. This is a 6. This is a 6. That has 5 carbons. This has 4. And we come back with 4 all the way through. CO2 is one of the ultimate outcomes of the reaction. If you take a look, there are 3 times that you create NAD+. For every 1 pyruvate, you will create an FADH2. Here's your energy. And then there's 1 more. We actually take and we produce a GTP from a GDP. Now, if you take a look at a textbook Most of the time, what we do in stoichiometries is we say how many ATPs we can make. Well, a GTP can transfer its phosphate to an ADP to make an ATP, and there's no loss of energy. And so we will formally make 2 GTPs, but in almost every table, we will equate them, and that's why they put it in parentheses, an equivalent of ATP. All right? It doesn't actually react with the A diphosphate and create ATP, the triphosphate. All right? So most of our energy is converted into those electron carriers. So we still have most of the energy sitting in electron carriers, both from glycolysis and from the pyruvate dehydrogenase complex and from the citric acid cycle, and really only a little bit of triphosphate intermediates were ever produced between these 2 cycles. And we've used up all of the carbons at this moment in time. Looking at the citric acid cycle, we've got a couple of important things in terms of the reaction. And so the acetyl-CoA will react with the first reactant called oxaloacetate. And the product will be, also water comes in as one of the reactions, which actually is a third reaction. So we have acetyl-CoA, oxaloacetate, and water, 3 reactants. There's 2 reactions here. All right. Now, we need to do some things here. When we look at it, the thing that we have to do is figure out where in the world things are and in terms of inventory. And so before you do any reaction, usually inventory is important. And so I will show you where things are. Okay? This carbon right here would be A. All right? This is the A carbon. This carbon right here is the B, and that's the B in the product. Turning around, this is a 1, and so this would be a 1. This is the 2, so here is 2. This one is 3, and I'm not going to put it there. I'll put it right here. That's 3. So this is the 3rd carbon, and then this one is 4, and that was the 4th carbon. All right? So what in the world is happening in this reaction? What is combined to what? I'm condensing, so I'm forming a new bond between what and what? 3 and B. Now, if I take a look at B, in the end, how many protons are there on that carbon? How many carbons are in the beginning? 3. Found an extra proton. If you look in carbon 3, you see at the reactant it has a double bond, and in the product it has a single bond. And the oxygen has gone from a ketone to a hydroxide. Okay? I actually add a proton on. And so, what would you say is the first step in a mechanism here? %80 of the time. What? We're going to deprotonate the acetyl-CoA. So remember, inside an enzyme, we always have a base, and we always have an acid. I will put the acid in no particular place, just kind of randomly on there. And so, which proton do I abstract? This one, or that one? This one right here? This one right here? That one. Doesn't matter. They're all equivalent. Just a matter about which one you feel. I feel that one better. Okay? then I now have formally a negative charge on this carbanion. Is that good? No, it's high energy. Why in the world would these protons actually be deprotonated? Is there an organic reason why those protons can be taken by a base? It's an alpha to a carbonyl. What I can do is create a resonance structure in which instead of the carbon being, the negative charge being on the carbon, I can resonate it up into the ketone. And so that lowers the energy. That creates the fact that those protons are acidic. All right. This negative charge is the reactive species. It will then attack at this carbonyl. Why does a nucleophile attack a carbonyl? Because it's partially positive, because the difference between electronegativity between the oxygen and the carbon. And when I attack, the electrons kick up. The electrons can be temporarily on oxygen, which is electronegativity, and so that's okay. Now, the 4 carbon and the 1 carbon also have ketones. Why are those not attacked? There are 2 reasons that you could tell me why they're not attacked. And it's better seen in how I drew them in the product. Yes, it's a ketone, but it's not just a ketone. It's a what? What is the functional group for A and 1? Oh, sorry. Well, no, sorry, for 4 and 1. They're not esters. They're acids. Okay? And I drew one of the resonance structures. The double bond is not just sitting there, it's resonating. And so it's not a true double bond. So organically, we don't attack an acid nucleophilically. The second and most important reason why in the world we attack at 3 is because, remember, none of these reactions are happening in an organic flash. They're happening inside what? The enzyme. The carbon of the acetyl-CoA is right next to that carbonyl, the way that they're bound inside the enzyme. All right, the last step in this particular mechanism, this step, is to form the hydroxide. Now, let me draw you the intermediate. Okay, this is carbon 3. I just put on this acetyl group. I just have this hydroxide. I have this acid. And I have this acid at the bottom. Is this the product? No. What's wrong with this? The CoA at the top. For it to finally react, I have to have a water addition. I'll deprotonate the water. I'll have a hydroxide attack at the carbonyl up, down. The CoA comes off as the leaving group, and I will have an acid. Now, the mechanism I drew, what would be the protonation state of the acid at the top from what I just drew? I attacked a what? I have a hydroxide, right? And so if you used all my arrows, you would actually have the product having a proton right there. But that proton its pKa is very acidic. As soon as it diffuses into the cytosol, that proton will be deprotonated because the buffering system of the cytosol will deprotonate it because of just a pKa argument. And so it ends up being, it becomes the deprotonated form and the citric acid itself. Now, one of the names of the citric acid cycle is the tricarboxylic acid cycle because we have an acid at the A, an acid at the 4, and an acid at the 1. Okay. Also, it kind of looks it, but kind of doesn't. There's symmetry here in the citrate. Okay, that's going to be important a little bit later. This molecule, although it's big and bulky, it is also symmetrical. All right, here is that intermediate where I'm going to have water come out and adding water. So dehydrate, and then we will hydrate back. All right, if I take a look, you can see that the extra proton is on the methylene group at the bottom of this slide. If I have an extra proton, a base in the enzyme is going to take it. I will then form a double bond, and the leaving group will be a hydroxide picking up a proton. That's where your water comes from. In the second, I'm going to rehydrate, and this is kind of weird, but I'm going to take a base, and I will take a proton off. I will have a hydroxide nucleophile that attacks here, and then it picks up a proton there. I go from citrate to isocitrate, and I've created an unsymmetrical molecule. Okay? Now remember, I said A, B, 1, 2, 3, and 4. All right, isocitrate. The next will be isocitrate to alpha-ketoglutarate. There are several intermediates in this, but what we're going to do is we're going to have a oxidative decarboxylation. Now, most of the time when we have an oxidative decarboxylation, what happens is you have two reactions. You have the oxidation and then you have the decarboxylation. Usually the oxidation happens first and then the decarboxylation occurs. The reason is we're going to create an intermediate that allows the CO2 to fall off. It'll be able to create a resonance structure that allows the CO2 to come off. And so, in this case, it is a dehydrogenase. I need an electron acceptor, NAD+. The mechanism will have a hydride fall off. I have two options, A and 2 protons. Those are going to be lost. Which one is going to be the one that is deprotonated, and which one is going to be the one that is the hydride that leaves? A or 2? All right. For those that believe it is 2, raise your hand. For those that believe it's A, raise your hand. All right. I've got 5 half-hearted A's and then a group that has, I have no idea. All right. It is actually going to be the A, because an intermediate that could be formed is a negative charge on an oxygen. The other one, if I took it off at 2, I would have an intermediate that could be a negative charge on a carbon. Negative charge on an oxygen is much better. Okay. But I will deprotonate. This will form the double bond, and that is the accepted hydride. If you wanted to prove that the 2 proton is the one that is accepted by the NAD, how could you set up an experiment in a laboratory that would give you definitive answer to that.
Is it using the absorbance at 340?
No, because if it took it off one or if it took it off A, you're still going to get the same product. But how could you tell if? How could you distinguish which one was taken off? This is a thinking question. This is, I don't know if this. There's nothing in my teaching that would have taught you this. This is just a real big thinking question. A resonance structure? A crystal structure. Oh, yes, that could, because you could actually see the catalytic base amino acid side chain. Which one is it closest to? Another method that you could do is you can create that molecule isocitrate, and you can label one of them with a deuterium and one of them with a proton. See if, depending upon where it is, if you put the deuterium on the carbon, is it found in the NAD, or is it the proton that is found in the NAD? And actually that was done to determine that the hydrogen on the carbon is the one that is accepted. All right, I have an intermediate, and it is now ready to decarboxylate, so I'm going to lose a CO2 in this step. Now, if I lose a CO2, that is simply the electrons fall in to create a double bond, and then this pair of electrons has to be the leaving group. Okay? In this structure, it will form a double bond, and then this will pick up a proton. There's a place for those electrons to leave and a reason. I create a very stable intermediate that doesn't have a lot of charge with it. If I actually tried to do the decarboxylation first, if I just went here, the only place that this pair of electrons could go is right there, a carbanion. That is really, really bad in terms of energetics. It can't go up and form a double bond because the only leaving groups would be hydrides or acid with an extra pair of electrons. And if it goes down, you might have a hydroxide fall off, but that's still high in energy if it's not stabilized. And so the oxidation allows for the decarboxylation to occur. Now, don't ever forget this, energy. All right? Now, to get to the alpha-ketoglutarate, the next enzyme in the process, the next structure in the process, I will take off this particular proton. I will form the double bond, and then now I have an acid that can be picked up to form alpha-ketoglutarate plus CO2. All right. In this case, the molecule is still asymmetric, and alpha-ketoglutarate we were going to see all of the time from this point on. All right. The next reaction is alpha-ketoglutarate to succinyl-CoA. All right. It is performed by the alpha-ketoglutarate dehydrogenase complex. Uh-oh. Have you heard of something that had a dehydrogenase complex? Pyruvate. And if you actually took a look at pyruvate, okay, pyruvate is different right here. It just has a methyl group there, okay? But it has an acid that is alpha-ketone. So it's called an alpha-keto acid. And what happened with pyruvate is we decarboxylated this and added CoA. And so an alpha-keto acid is converted into a ketone that has an acetyl group. This would be exactly the same mechanism. It requires TPP, it requires lipamide, it requires FADH2, it requires NAD+. and has the same mechanistic steps. All right, and we lost a CO2 in that. It's an oxidative decarboxylation, and again, energy is produced. Okay. Now, at this moment in time, the acetyl-CoA came in with two carbons, and we've now lost 2 carbons. The 2 carbons that are lost are not the carbons that came in, because if you continue to number them, we still have the 2 that came in from the acetyl-CoA. We have a net loss of 2, but not an actual loss of 2. Now, I won't go through this mechanism because it's long and we've printed it out. All right. In this particular case, it's kind of an interesting reaction. The succinyl-CoA, all right, will react with a PI that is not drawn on this slide. I have no idea where the PI went. And so if I actually draw that out, a PI in its normal protonation state within the cell has 1 of the oxygens protonated and 2 negative charges. One of the negative charges will actually be a nucleophile, and I will attack, kick up, kick down, and the acetyl-CoA falls off. This is an interesting reaction. We've seen this before in glycolysis, in which I phosphorylate an intermediate without use of ATP. So this is really a place where energy is invested. And then this is a abbreviation of GDP. A GMP plus 1 more phosphate is a diphosphate. And then this will also act as a nucleophile. It will kick up, it will kick down, and then one comes off is an acid called succinate. But energy is created. And most people will talk about this as an ATP equivalent. Okay, got a star. Succinate. It is symmetrical. Once the succinate leaves the enzyme, which is succinyl-CoA synthase, it floats to the next enzyme. Okay. In this particular reaction, you can actually see that the enzyme is catalyzed and becomes covalently linked as part of the reaction. So the phosphate, before it is transferred to the GDP, actually gets transferred into the enzyme and placed on a histidine as the actual intermediate. Succinate is a leaving group. The only problem with the artist in their way that they drew this was that the electrons have to kick up and kick down there. Electrons have to kick up and kick down there. And so the artist may not have a organic chemistry degree and kick up and kick down. But you see that written quite often in texts because it just reduces the number of arrows that they draw. Okay. When succinate falls into the active site of succinate dehydrogenase, okay, it's symmetrical. So it can orient itself one of 2 ways. If you were keeping the numbering of the molecule at this point in time, when succinate comes out of the enzyme and enters into this enzyme, this carbon could be that A carbon, or it could have been the 4. I believe it's 4. Nope, the 1, the way I numbered it back. And it could be this or 1. Now, how would you represent it? Well, 50% of the time, the top one would be A, and 50% of the time, the top one would be 1 at this moment in time. That's as good as you're going to get. All right. FAD is an acceptor of a hydride from a reaction just between alkyl chains. So a base could take this one, fall in, accepted by the FAD, and then the FAD has to pick up a proton. And which one is taken is really dependent upon how it enters into the enzyme. So they're equivalent, either the top or bottom, because we can even, the numbering system, we couldn't distinguish them. And we're at fumarate, which is still symmetrical. The last set of reactions, we're going to start to create asymmetrical. Water plus fumarate and fumarase. We have a base will deprotonate the hydroxide. The hydroxide will attack at this carbon because the enzyme dictates that's where it will react. And then I'll pick up a proton to get the L-malate version, because now we have stereochemistry. And then the last reaction, oh, wait, I just wanted to, we need to go back one more. Energy is produced. And then the last reaction, L-malate, will be converted into oxaloacetate, which was one of the original. A base will take this proton, form the double bond. The hydride is accepted by NAD, and we've extracted more energy, our last amount of energy. And we have recreated the original reaction. The overall reaction, this is something that you would really need to know. An acetyl-CoA comes in, 3 NAD pluses, an FAD, a GDP, 2 waters. I will get 2 CO2s off, 3 NADHs, 1 FAD, a GTP, 2 protons, and CoA. This is stoichiometry for 1 acetyl-CoA. If you go all the way back to glycolysis, all of this would be doubled for 1 glucose. Okay. Regulation of this, things that make it go, things that make it stop. All right. Calcium is in there again. Calcium, again, is an indicator that muscle contraction, and so make these go. It tends to be this. If you have the reactants in high concentration, NAD+, CoA, ADP, which is low energy state, it turns it on. And so you see these kind of things here, a reactant, low energy, makes it go, low energy, makes it go, an indicator of low energy, makes it go. The products, NADH and acetyl-CoA, if that builds up at very high concentrations, it actually will stop the production of pyruvate to acetyl-CoA. Lots of energy, ATP will stop it. Product stops it. Citrate, a feedback, stops that enzyme. ATP, high energy charge, stops that enzyme. And another feedback, if I get the succinyl-CoA coming back, it'll stop this process. NADH, a product, will stop the process. ATP, high energy. That's no problem. Succinyl-CoA, feedback, that seems okay. ATP, that seems logical. And then an enzyme that we didn't talk about, we can create acetyl-CoA from pyruvate, and we'll talk about that later in a different reaction scheme. This will be part of gluconeogenesis, and this becomes important in starvation states. Okay. The alpha-ketoglutarate dehydrogenase complex we already talked about. The only difference is the R group on the carbonyl, and they go through the same reaction mechanism. So that I've already talked about that. Now, when you talk about that reaction for those two dehydrogenase complexes, we can talk about a disease. And beriberi is a disease because of the deficiency of thiamine, which is vitamin B1, which is the component that makes the reactive TPP. What happens is pyruvate concentrations go high and alpha-ketoglutarate concentrations go high in the blood. Okay? And what happens is when you get a high concentration of an intermediate, almost always you get issues with the signaling of your nerves. And so in this case, the nervous system, the peripheral, and you have problems with control of your muscles. This tends to be a serious problem in the Far East because of this fact. They eat a lot of white rice. The vitamin B1 is found in the husks. And so what they do is they get rid of that, and they don't, they have a lot of caloric intake, but they lack this one vitamin. And so when you polish the rice. Here is a thing that people really get mixed up. When you have a disease, you have two different things. You have a symptom and you have the disease itself. In this case, if you have high concentrations of those two intermediates, that is not the disease. That is the symptom. Those are the things that you see in the blood work. What the disease is a misfunctional enzyme, or a couple of them, because you don't have the cofactor. You have a lack of vitamin. And so don't get the symptom confused with the diseased state. Okay? Citrate synthase reacts acetyl-CoA and oxaloacetate. Oxaloacetate has to bind first to actually create the binding site, so it has an ordered binding, and it actually places the enzymes in the orientation that it needs to react. We've had deprotonations, and here I show you that the aspartate 375 is the actual one that gets deprotonated. And then the attack, the proton actually comes from histidine 320 and the mammalian enzyme. Again, that's not as important. You can identify the source as long as there's a proton. We've been saying it comes from an amino acid, and here I articulate it, but you don't have to remember it's 320. Okay. The last step, we actually have the water come in and take off the CoA. We've already looked at that mechanism. Okay. This molecule can be written in lots and lots of different ways depending on upon what textbooks you look at. What is unfortunate that you can't see physically is that citrate is actually a symmetrical enzyme, a symmetrical molecule. It has symmetry right here where this is resonating, and above and below I have two acids, methylene and acid. It actually is a symmetrical molecule. We talked about when succinate comes out, it's symmetrical, and I start losing the order of where things are. All right, but the citrate synthase itself, actually the way it bonds, doesn't lose its orientation. What that we've done is a lot of experiments where if we labeled the A methylene and the B methylene, the A methylene is the acetyl-CoA coming in, the B was from the oxaloacetate. If we look at the citrate and then going to isocitrate, well, citrate is a symmetrical molecule. And if I take off water, well, I could take it off the top or the bottom, or it could fit in both ways. Is the carbon at the bottom with the hydroxide, is that A, or is that B, or is that a mixture? And what we found surprisingly is that the hydroxide that is on that carbon is always on the B, or the carbon that came from oxaloacetate. Never do we ever see that hydroxide bound to the carbon that came in with the acetyl-CoA. Okay, and this has actually been followed by carbon-14 labeling. And so they labeled carbon-14 and the acetyl-CoA and allowed the reactions to occur, and the carbon-14 was always in the top, furthest away from the hydroxide, never next to the hydroxide. Well, how in the world is that? Well, that is symmetrical, yes. And if you did the reaction in organic flux, you would get a mixture. What the stereochemistry comes in is not because of the stereochemistry of the reactant, it is because the enzyme has stereochemistry. And the reaction is facilitated by how it binds inside the enzyme and coordinates to an iron center. And so when you create negative charges as part of this reaction, the negative charges are stabilized by iron centers. And it will always coordinate in such a way that the B carbon is the one that's going to be deprotonated in the reaction. The A carbon always sticks out of the active site. And so we always have that carbon coming in furthest away from the hydroxide, which gets converted into that ketone. And so we keep asymmetry even in that particular citrate, which is symmetrical. Succinate doesn't have that same luxury. Succinate does change. And that's important later, especially when we track things with radioactivity. All right. NAD+. When we actually use NAD+ as an electron acceptor, we can have a hydride accepted. In this case, a deuteride is accepted because we use a deuterated reactant. And so the NAD will accept a hydride, in this case the deuteride, and so I can distinguish which proton was originally there and which one was accepted. What is really strange is this. If I do that reaction and I load NAD with a deuteride, and it becomes now, we call this NADD, if I take that, isolate it, and react it with something else, you always have the hydride that comes in is the one that's donated. It's kind of an interesting reaction because that flat ring is symmetrical and either one could have been donated back. Citric acid cycle, yes, creates a lot of intermediates, and I'm creating lots of energy, but these intermediates will then be used to synthesize lots and lots of things. Mainly, and you can see just about everything. I can make carbohydrates, I can make proteins, and I can make fat, all from the citric acid cycle. All of those arrows going in those directions, as I show here, can actually have a reverse direction in the slide, and we'll see that in a little bit. Okay. All right. The last thing that we can take a look at is the reaction. The first thing that came in was acetyl-CoA plus oxaloacetate. Okay. That is great, and that's a great beginning, but here's a problem. If by chance you start doing synthesis, and you take off some here, and you take off some there, and you take off some there, and you take off some here, what you can do is deplete the citric acid cycle. And then you have none of that. If you have none of that, you can't put acetyl-CoA back into the system. And so we got a problem. I can run out of oxaloacetate. So the body has the ability to take part of the pyruvate and make acetyl-CoA in the way I showed you. It can also take part of the pyruvate and make oxaloacetate so that it can refund the structure that might be deficient. To do that, pyruvate will react in an enzyme called pyruvate carboxylase, and it will react CO2, pyruvate, ATP, and water to get oxaloacetate. Now, here's the problem, okay? I say pyruvate plus CO2. Look at the first slide up at the top. I see ATP, and I've got that in the list, and I see something else. What do you see up there? What is the reactant in the figure? Is it CO2? Can you read it? What is it? What is the ATP reacting up in that first place? Bicarbonate. Okay. Well, I said it reacted with CO2. Well, CO2 is a gas, right? Gases don't dissolve in water very much. Okay. But inside our bodies, CO2 reacts with water. The outcome is this molecule, which has never been isolated, which immediately breaks down into this ion, the bicarbonate ion. So when you see bicarbonate, it can be distinguished or it can be explained as or outlined as CO2. So CO2 and bicarbonate are equivalent items. They get interconverted, and the CO2 bicarbonate system is the major buffering system in all of biology. So they're roaming around. The CO2 gets activated by adding a phosphate onto it, and that CO2 then is carried by a carrier. In this case, it's called biotin. And that CO2 then reacts with pyruvate to create oxaloacetate. Pyruvate is a 3-carbon intermediate. Oxaloacetate is a 4-carbon intermediate. And what you see here is pyruvate is an acid, a ketone, and a methyl group. Well, I take off a proton and I add a CO2, and now I have oxaloacetate. And that's where we can fund oxaloacetate so that it can be reacted with the acetyl-CoA. All right. In plants and bacteria, there is a process that is similar to the citric acid cycle, but different. We eat and breathe in oxygen, and our waste products are water and CO2, right? Plants, what is their waste product? What do they breathe in, and what is their waste product? I'm going to Bio 110. They breathe in CO2, and they breathe out oxygen. So how in the world could you have a citric acid cycle that's putting off all CO2s in plants? Well, it's got a modified, something that is very similar to the citric acid cycle. What happens is acetyl-CoA can come in, and it can react with oxaloacetate to make citrate, just like ours. So that looks just like the citric acid cycle. And then it can make isocitrate, just like our citric acid cycle. But at this step, we have a different enzyme that will have succinate come off, and I get an intermediate that is different than the citric acid cycle. And then another acetyl-CoA come in, and it reacts to make malate, and the malate to oxaloacetate is exactly like ours. We allow acetyl-CoA to come in, just like the citric acid cycle, and we have many of the transformations, but we have no CO2 coming off. All right? In this process, we make energy. Now, granted, it is not as much as our citric acid cycle, but this is a little bit of energy. This cycle is used when the seeds are germinating. Because when a seed is in the ground, there's no light, there's no photosynthesis going on, but it needs to grow. The seeds have fat in them. Fat will be broken down into acetyl-CoA. So the seed will utilize the fat stores in the seed until it can grow enough out of the ground so that it can create chlorophyll and start absorbing from the sun and start the normal photosynthesis. So this particular process is intended to give plants their energy until they can draw it from the sun. What I'd want you to do for that is to compare and contrast citric acid cycle, know what's similar and know what's different between the two cycles. All right. I think that's it. And also know that that process is used when the seeds are germinating. That's really the big piece of why we introduced that particular idea. Here's the quiz for you to work on. And the reason why we do this is because this is actually one of the ways they discovered the cycle itself. If a glucose, way back up in the front, glucose 6-phosphate is labeled with C14, and I'm going to label it in carbon number 2, and you let it go through glycolysis and the citric acid cycle, what I want you to do is to tell me where the CO2 ends up once you get to oxaloacetate. Okay? And so it's in the 2-carbon of glycolysis, and it's going to go all the way through, and you're going to tell me, is it in one of those atoms in oxaloacetate, or could it have fallen off as a CO2 and not even in that molecule, or is it in multiple places on the oxaloacetate?